Cells and Stem Cell Niche session. I'm George Consarelis, and I want to welcome you to, I think, what will be a very exciting session. There are a few changes in the program I wanted to mention. One is that Colin Yehoda couldn't make it, but Claire Higgins stepped in to take his place. She's a lecturer at the Imperial College in London. And Manabu Oyama is a professor at Kyorin University in Japan, and he's a co-director as well. And then one of the speakers, from France, Natalie, unfortunately had to cancel because of the terrible events there. And what we've done is we've increased the length of the first two talks by 5 minutes each. So I have potential conflicts, but not relevant to this session, especially since I'm not speaking. And we had the learning objectives are to describe some of the latest developments and research on hair follicle stem cells. And very excited about this program because these individuals really represent the future of dermatology and hair research. Dr. Tumbar is from Cornell. She's done seminal work developing a mouse model that's been used by many other groups, the H2B GFP mouse, that's been very powerful. Dr. Rompalis is an up-and-coming investigator who's at the University of Pennsylvania. Michael Rendell is also at the cutting edge, very exciting talk, looking forward to it. And Jerry Lynn is giving another talk and will dazzle us as well. So I think it's going to be a great session. Without any further, I just want to remind you that there's only that guy is the only guy that's allowed to take any pictures. No one else is allowed to take pictures or video. So we'll get started. So here we go. Dr. Tumbar, otherwise known as Goina, will give us a 25-minute talk.
Hello, everyone. Thank you for coming to this session. I also like to really thank George for inviting me. It's really exciting to be here, and I'm very happy to be part of this. I worked on hair follicle stem cells for about 15 years, and what moves me are really basic science questions related to what makes a stem cell a stem cell. And today I'm going to focus to one aspect of the research in my lab that really looks at how a single gene can coordinate various different aspects of hair follicle development and of hair cycle, and does that by really modifying both the cell intrinsic properties of stem cells as well as by remodeling the environment around these stem cells. And when I started working in this field, a lot of the talk was about what really makes a stem cell, and there were two main features that might be at work. One was that there might be some unique intrinsic features of these cells, such as specific transcription factors, also known as core transcription factors. And we actually know that in embryonic stem cells that is indeed the case. We know the 4 factors, 4F, that are really responsible for the stemness of those cells. For tissue stem cells, we really it's not really that clear what these intrinsic features might be. I just listed a number of them right here. But it's also possible that the environment in which these tissue stem cells reside is really unique, and that's what's guiding them and making them so special. So when I started working in this field, I was really inspired by the work of George Consarelis, who used label nucleotide to do a pulse chase in the skin and find cells that retain label, these label-retaining cells, as putative stem cells in the bulge of the hair follicle. And I followed up on his steps and decided to take a little bit of a different approach to do a pulse and chase with a histone fused with GFP, using a transgenic model in which the expression of this gene can be turned off via doxycycline. And then the cells that rapidly divide will dilute the label, and infrequently dividing cells, which are concentrated or clustering in the bulge, can be labeled that way, and then that allows us to FACS sort them, use fluorescence activated cell sorting to isolate the cells and do molecular profiles to look at the genes that are expressed there and get back to these questions of what makes a stem cell a stem cell. Can we learn from the genes that they express what makes them so special? And as I put together this talk, I just realized that we wrote this review back then in 2004 because we were very intrigued to find that amongst the genes, many of the genes that we found upregulated in the bulge labor-retaining cells were genes that seemed to be responsible for signaling to the environment. And so it gave us this idea that perhaps the stem cells themselves might organize the environment around them, and they're not always the ones that receive instructions about what to do. And there were so many leads and so many genes, and there were so many connections with nerves and with the dermal sheath around the hair follicle, and with perhaps blood vessels and other components. And we didn't think about the fat then, but we know from Valerie Horsley now that they are also part of this environment. And it felt like this is a very gargantuan task that was pretty much impossible to take, to look at all these genes and then prove this model. So I decided to step back and really focus on what I thought would be the cell intrinsic differences between stem cells and their more differentiated cells. And so I wanted to look at genes that might be responsible for stemness in different tissue stem cells. And that's how I chose this gene called RUNX1, because it was previously shown to be essential for hematopoietic stem cell emergence, important for proliferation of neural stem cells, and we found it expressed in the hair follicle stem cells. And there are various reasons why I picked RUNX1 besides this. It's a very conserved transcription factor, so it's likely to be a very important factor. Again, it was important for other stem cells. It's involved in signaling pathways such as Wnt and BMP, which are important for hair development. It was shown before to be expressed in whisker follicles by immunofluorescence. A mutation in RUNX1 DNA binding sequence is associated with psoriasis in human. And last but not least, there were a variety of genetic tools that allow us to really look at the function of this gene in mouse skin. And I listed them here at the bottom, and I'll show you how I took advantage of these tools to understand what this gene is doing. So before I move on into the data, I just want to give you a bit of an overview of what we found about this gene, because it's actually important in various places in development, and it turned out to be expressed in a very interesting pattern that we didn't even predict based on our initial experiments. And I'm going to summarize here pretty much the whole past 10 years of work with 4 or 5 papers published on this gene to tell you that it's expressed very early in embryogenesis in the hair placoid, and if you knock it out in the epithelium at this stage, impairs the timely hair follicle formation. So hair follicle formation is delayed. It was also expressed in this region underneath the hair follicle, in the dermis, very transiently, but if you knock it out in the dermis during those times, initially nothing happens, morphogenesis occurs normally. But if you then look at the adult hair follicles, these follicles fail to be maintained, they turn into these huge sebaceous glands and disappear. We then also found that it's expressed very strongly at the transition between anagen and catagen, and we show by forced expression of this gene during this stages that it's in fact sufficient for catagen onset. It is not necessary, suggesting that there are other genes that back it up when catagen is to be instated, but it is in fact sufficient. Third, and the most important, which I'll focus most of the last part of my talk, was that this gene was expressed in the hair germ here at the base of the bulge during the telogen to anagen transition. And if we knock it out in the epithelium at that stage, it delayed the anagen onset. So the hair follicle growth was delayed. And finally, it's expressed at relatively low levels during the early anagen phases when the bulge stem cells self-renew. And if you knock it out during those stages, then proliferation is mildly impaired. So now I'm going to show you some of the data that backs up all these studies. Here at the top, I'm showing you a lag-Z knock-in into the RUNX1 locus. So wherever you see blue is where the RUNX1 is expressed, and here is expression in some cells of the hair placoid. A few cells here at the top of the hair germ. Here's that expression in the mesenchyme that I was telling you about, very strong. We backed it up with immunofluorescence staining, shown here in red. During adulthood, again, it's expressed in the hair germ very strongly and at the base of the bulge. Again, backed it up with immunofluorescence staining. And we also were fortunate to have these tools where the Cre-ER is knocked into the RUNX1 locus, and this genetic tool allows us to do lineage tracing. So we mark genetically the cells that express RUNX1 and look at their fate, and we could do that by injecting tamoxifen either early during embryogenesis or in adulthood, and whichever way we looked at it, we found that cells that express RUNX1 in the epithelium contribute to all the lineages of the hair follicle, and they do that repeatedly, demonstrating beyond that RUNX1 is expressed in the stem cells of the hair follicle. And last but not least, we found that RUNX1, which is part of this family of cancer genes that are important in various cancers, especially RUNX1 is important in leukemia, we found that in the skin, cells that express RUNX1 can be at the origin of epithelial tumors. As you can see here, this epithelium of this tumor is all blue, suggesting that it comes from a cell that expressed RUNX1 at the time of induction. So then we went on to do functional studies, and the first part I'm going to focus on the loss of function studies is when we knock out RUNX1. And we're able to do that in various compartments of the skin differentially. So we started by knocking it out in the epithelium using the K14-CRE or K14-CRE-ER. And we found that in the absence of RUNX1, first there's a delay in hair follicle morphogenesis that's then eventually caught out by, so eventually the hair follicles form. And later on in adulthood, there is a delay in anagen onset, and that's what's shown in here, while the wild-type hair follicles enter anagen and grow new hair shafts. The knockout follicles stay in telogen for a prolonged period of time, and then eventually they start growing. And we found that during embryogenesis, Wnt signaling was down in both compartments when we knock out RUNX1 in the epithelium, and that could explain the phenotype of the embryos, because we already know from Sarah Miller and other people in the field that Wnt signaling is very important for this step in hair formation. We also went on to knock out RUNX1 in the mesenchyme to see what its role is, and as I mentioned earlier, we found that first, morphogenesis is not affected by knocking out RUNX1 during the embryogenesis in this stage, where it's very transiently expressed in the dermis. But what happens is that, in fact, Wnt signaling is upregulated now in both compartments, and we have really high levels of LEF1, which is part of the Wnt signaling pathway, expressed throughout the skin. And so, therefore, it was not surprising that as the follicles matured, instead of continuing to form hair follicles, they formed this big sebaceous glands, and then eventually the whole hair follicle was degraded. And I'm saying it's not surprising because it's already known that if you put LEF1 at high levels in the hair follicles throughout development, that's exactly what happens. And finally, in the tumors, if we knock out RUNX1 in tumors, either before the tumors form or after, we see effects. So if we do it out before the tumors form, we don't get any tumors. So if we knock out RUNX1 before we induce tumorigenesis, we don't get any tumors. And if we knock out RUNX1 after the tumors form, the tumors shrink. And we think that's really related with regulation of STAT3 phosphorylation through these direct target genes of RUNX1, SOX3, and 4. And all this work was published, and I'm summarizing it here very quickly. And all these genes here downstream of RUNX1 explain the phenotypes of the embryogenesis, the mesenchyme, and the tumorigenesis. But what remains unexplained was how does RUNX1 work to promote hair growth, to promote anagen onset during adulthood, and why is it that when RUNX1 is missing, this is delayed? And this is just to show that we also look at human skin for the tumorigenesis. So the rest of my talk is really going to focus on this adult phenotype, and I just wanted to show a picture of the mice for those of you who might not be so used to look at skin sections, what I just showed before, the phenotype. What it looks like is that if you shave mice very carefully so you don't injure them and induce hair growth that way, the wild-type mice will grow hair after a short period of time, while the knockout mice remain without hair for an extended period of time, and then they eventually grow it back. And so that's what the loss of function phenotype told us about RUNX1 function, and we wanted to see whether we can learn more about this from the gain of function phenotype, which is we want to now put RUNX1 in the bulge at high levels whenever we want to and see what the effect of it is, and perhaps that will teach us more about how it's working in inducing hair growth. So we made a transgenic mouse in which RUNX1 expression is turned on in the epithelium at levels that are high, but they're comparable with endogenous levels similar with this expression of RUNX1 in a small fraction of inner root sheath cells that you can see here in the hair follicle. And here on the right is the expression in the transgenic mouse that was induced with doxycycline to express RUNX1 for 4 days. So the expression is high, but it's relative similar with the high levels of endogenous expression, so it's not tremendously high. So just to start off by saying that the first thing was to ask whether, since RUNX1 is important for hair growth, is necessary, is it also sufficient for hair growth? And so what we did is to turn on RUNX1 expression at the second telogen when you have very long period of quiescence and see if it's sufficient, and the answer was no. It was not sufficient on its own to induce hair growth. Before I move on, I want to have you look a bit more carefully at the expression pattern of RUNX1 during this stage of the first hair cycle. So again, it's expressed highly in the hair germ here and somewhat at the base of the bulge. It's in this inner root sheath cell a bit later, but watch how it's really rapidly downregulated in the matrix at these early stages. And then it becomes very strongly upregulating during catagen, and throughout catagen, it's very highly up in this CD34 positive cells, which are the stem cells that are migrating out of the bulge at this stage, and then it remains high just in the hair germ over here. And perhaps it's not surprising that RUNX1 goes down very quickly here, and I'll tell you why in a moment. We looked at this a bit more carefully with H2B-GFP pulse chases and found that, in fact, as soon as these hair germ cells begin to divide, they downregulate RUNX1. And in fact, if you put RUNX1, force it to be expressed in the matrix during these stages, it induces very rapid apoptosis. And so if you put it up here or down here, whichever of these early anagen stages you put it on, it will induce very rapid catagen. So what this told us is that, in fact, RUNX1 is important for anagen onset and proliferation, but it does that prior to proliferation onset. It's somehow preparing probably the hair germ cells to be activated and proliferate very quickly in the following stage. And so we were thinking that perhaps RUNX1 is expressed in these precursors of the hair germ cells, and we wanted to look at that carefully using our lineage tracing tools. So we managed to address this question, trying to mark these cells here that migrate from the old bulge and make the new germ. And actually some of them were shown by Elaine Fuchs to return to the new bulge and stay as stem cells. And we know that RUNX1 is expressed in these cells right here at the protein level. And we can use our RUNX1 CREAR to mark these cells, along with these other cells here in the inner part, but these cells will be pushed out of the skin surface and they die during telogen, so they won't survive. And so these cells from the outer root sheath and the old bulge are the only ones that survive. And the old bulge was marked, but very sparsely. Very few follicles showed the old bulge being marked. And so the cells that we're looking at here that survive are cells that express RUNX1 in this trail here below the bulge. And indeed, we find that these cells contribute to both the hair germ as well as the new bulge. And so we're thinking that perhaps if you are a bulge cell that starts expressing RUNX1 moving out of the old bulge and become a hair germ, while you're expressing this gene, you become a hair germ cell that is actually ready to proliferate, and that RUNX1 plays a crucial function in this. And so the very obvious thing to do is to try to see what are the target genes of this transcription factor, how is it doing that, and how is it doing that. And so indeed, when we look at microarrays that are one day after induction of RUNX1, we found that the bulge signature genes are very heavily downregulated within the bulge cells that overexpress RUNX1, while the hair germ signatures were very heavily upregulated, while what we would think of as matrix was more neutral. So we think that indeed RUNX1 expression start pushing the bulge cells towards a hair germ phenotype. It also does that functionally, because if you induce RUNX1 at early stages of anagen, the bulge cells proliferate faster, which is what you would expect of a phenotype that's more consistent with the hair germ cell, and they downregulate CD34 along with all the other bulge markers that we found right here. And actually, if you wait for a while, you find that the bulge cells eventually cannot sustain hair growth consistent with the fate of progenitor cells. So these cells that express RUNX1 are now shorter lived, they cannot keep on going, so the hair follicles are lost. And also by colony forming assay, we showed that if cells that overexpress RUNX1 proliferate rapidly and then they senesce, while cells that don't overexpress RUNX1 proliferate slowly, but they keep going. And so this rapid proliferation followed by senescence is again a phenotype of early progenitor cells, which is characteristic of the hair germ. So we then wonder, wow, that's interesting, and is this reversible? Because if you express RUNX1 in these cells that then return to the bulge, it should be reversible. And so what we did is our tools allow us to express RUNX1 for a few days, make sure that changes occur, and then remove it for 20 days and wait and see whether these phenotypes are reversible, and the answer was yes. And this is work that we published last year in Cell Reports. And so what we think RUNX1 is doing is that it's expressed in quiescence in bulge cells that are migrating out of the bulge. And as soon as they're out of the bulge, they start expressing high levels of RUNX1 and start their path onto becoming an early progenitor hair germ cell. And if they remain expressing RUNX1 there in the hair germ, and there's an inductive signal, antigen signal, that's coming in, these cells will proliferate very rapidly downregulated RUNX1, otherwise they'll die. They cannot tolerate it, and become matrix cells. But if they, in fact, return to the upper new bulge, they will lose RUNX1 expression and they will stay a stem cell. And this return also seems to happen when the hairs are plucked, as shown by George Kosterellis a number of years ago, that the hair germ cells will go back and repopulate the bulge. So how is RUNX1 doing all this still? Like, what are exactly the genes that are regulating these transitions, and what can we learn about the various mechanisms that are at play? And if you look at the microarray of RUNX1 transgenic, there are thousands of genes that go up and down within just one day. So there was really a lot happening that's just driven by one transcription factor in these cells. And I'm going to list a few that we are very excited about and we're following up on its leads. One that was the most obvious at the beginning was that there are this modulation of cyclin-dependent kinase inhibitors. In the RUNX1 knockout, these are upregulated, and in the RUNX transgenic, they were downregulated. And the most prominent one was P21. We actually published a paper on this. I'll show you a little bit of the data if there's time. I'm running a bit out of time, but the short story to that is that P21 was not the answer. Then there were a number of genes that suggest that RUNX1 might be remodeling the environment, such as the blood vessel, the nerves, and the dermal sheet, and perhaps these things are acting together to promote hair growth. There are genes that suggest that RUNX1 might be important in migration and rearrangements of the stem cells in the niche. And then finally, that RUNX1 might regulate lipid metabolism through various genes that are important for this. So this is the study where we looked at P21 and whether upregulation of P21 could be responsible for, in cell culture, for cell growth defects. And we found that indeed in cell culture, P21 can rescue the RUNX1 phenotype. But what we found was that in vivo, in fact, knocking out P21 in conjunction with RUNX1 not only did not rescue the hair growth defect, but accentuated. This is the RUNX1 single knockout. It's delayed in its hair anagen onset about 1 week, while the double knockout is way delayed, 5 or 6 weeks. And we have some hand-waving explanation, and we're trying to follow up on this. But the bottom line for this story is that it wasn't necessarily through just the cyclin-dependent kinase inhibitor regulation. So then to show you a bit of the data from remodeling the environment, this is all unpublished data that we're really excited about, is that we started looking at blood vessels in transgenic knockouts and wild types, and we found that the transgenic skin shows more blood vessels that are more in contact with the hair follicle than the wild type, while the knockout showed the opposite effect. So RUNX1 is modulating angiogenesis around the hair follicle. We quantified that. We also look at neurons, and we found the opposite effect, which is that while the wild types have a certain amount of neurons around the bulge, the knockouts have way more, and it seems like the neurons are really glued around the hair follicles, and the transgenic do the opposite. So RUNX1 is repelling the nerves away from the hair follicles, and it's also organizing the dermal sheath around the hair follicle, as seen with this vimentin staining. We've done some migration assays that I'm just briefly whipping through in the interest of time, that in the absence of RUNX1, cells don't migrate well, and we think that impairs somewhat the migration of cells out of the bulge during this early, during catagen. And finally, we looked at lipid metabolism, and if we felt that it really makes sense that RUNX1 might be important in metabolism because it's expressed during this quiescent phase prior to activation and proliferation. Perhaps what's doing in the hair germ cells is help them grow so they're ready for proliferation. Or perhaps a more interesting possibility is that it's remodeling the membrane so they can be more susceptible to activation signals, proliferation signals. And we think we have some data that starts leading into that. These are genes that were from microarray that are important in lipid metabolism, and many of them are genes that induce double bonds in fatty acids, in actually phospholipids, which are part of the plasma membrane, and the composition of the plasma membrane can be important for signaling. And so one of the ones that we really spent more time looking at was SCD1, and we also looked at phospholipid composition, both in the transgenic as well as in the knockout, when we found that was changed. This is our SCD1 data where when we overexpress RUNX1 in cell culture, SCD1 goes way up. And in the tissue, we see that SCD1 seems to be down in the hair germ in the knockouts. And this is our membrane organization, or fluidity assay, where we look with fluorescent dye at how membranes behave, how this dye is interacting with the membranes. And we found that in the RUNX1 knockout, the membrane is perturbed, and we're trying to figure out whether this perturbation has to do in any way with signal transduction, and that's how far we went, where we got to. And so the take-home message, I won't go through all the things, or maybe I should have a minute, 30 seconds. What I showed you is that RUNX1 controls the hair follicle stem cell to early progenitor transition, and the subsequent timely activation at anagen onset, that these mechanisms are pleiotropic and are both cell autonomous and non-cell autonomous, and that RUNX1 modulates both cell intrinsic and environmental factors that we think are coordinated in a way that is responsible for its phenotype. And so going back to our review from 2004, I felt like I was really fortunate to start working on a transcription factor that initially I thought it's just going to work at the cell intrinsic level, to find that in fact this factor is working both on the environment as well as on the cell intrinsic characteristics of stem cells to regulate its activity. And this is a picture of my lab, and many people contributed to this work, and we have quite a few, 4 or 5 papers published. Most importantly, I'd like to acknowledge Karen Osorio, who is actually now working at B&G, and she was really seminal in starting this project. And I'd like to end by putting a plug for a conference that I started, a Gordon conference on tissue issues and resident stem cells in adult epithelial. And we have a hair follicle session there that's chaired by George Gotzarelis. And I hope that you'll find time to come. It's in August 2016. And I really hope to see some of you there. And with that, I'd like to close and thank you for your attention. Just in time.
We have time for some questions. You know if it's expressed in human hair follicles?
Yes, it is expressed in human hair follicles as well as in the same pattern as in the mouse. With help from Ralph Koss, we figured that out. And it's also expressed in human squamous cell carcinomas of both oral epithelium and skin.
Valerie.
Okay.
Like two?
So the migration, I think, is really interesting, and something that I think, and as the field, we haven't really understood how these cells are migrating around. And so I'm curious what you think about how they are doing this in terms of adhesion to the dermal sheath or to other cells, and how does RUNX1 control those processes?
I think in the vivo there are multiple things happening. So there are many things in the ECM that seem to change, the integrins seem to change, so there are many things like that change. I think what you might be getting at is how this migration is even possible through this epithelium. And I feel that, I thought about this a lot, and it seems like it's very interesting that it occurs just at catagen onset. And it could be that what happens is the non-bulge cells begin to die and liberate spaces along the basement membrane, that then the bulge cells, which are actually very crowded in the bulge at early anagen, they proliferate and they stay in the bulge for a while, so they're extremely crowded. And now having spaces along the membrane because of the cell death, they begin to leak out. And I feel like what happens also is that by doing that, they lose contact with that inner layer of the old bulge, and that might be what's turning on RUNX1. But RUNX1 is stimulating migration even in the absence of all these things. So if you just do it in cell culture, you will see it. So I think that there are multiple things at play.
Bruce?
So when you do the TED-induced RUNX1, you're driving it in a lot of different cell populations. Is there a difference in which you have a signature of response genes? Is that specific to bulge, or is that sort of generally expressed in all the keratinocytes? Did you look
the signature which is changed. So there are different changes in the hair germ relative. We separated the bulge and the hair germ, and the bulge was the one that really made sense in the way that we see downregulation of the bulge signature and upregulation of the germ signature. In the germ, it wasn't the same thing. It seemed to accentuate more the hair germ genes. And in the basal layer, again, there are other changes. And this isn't surprising because RUNX1 is already known to play different roles in different cell types. So it's really context dependent. So we think that that is the case.
and I think you mentioned this, but in the knockout, the secondary germ still forms pretty normally.
It does. It's just not activated in time. Yeah.
Thanks.
Okay, thanks very much, Dorina. Oh, is there none? Sorry. Real quick, one more question.
Oh, just the SCD1 story reminded me the Acebia mouse that doesn't have any hair follicle and has hair loss, and the knockout also has hair loss. I wonder, when you overexpress that in the cells, and you mentioned that all the genes that you had in the panel, the end result is synthesis of triglycerides. The very end results, they are all lipogenic. Do these cells, if you let them go, they become more like SIBOcytes because they do come? No, they don't. Okay.
Thanks. Thanks. Thanks.